Hello, sir. I am Aayaam Jain, and this is my submission for round 2 of Iron YPT. The topic I have chosen is Droplet Microscope. So let's begin. In round 1, via an experiment, I had shown that if I take a water drop placed on a plastic slide, it can magnify the object, for example, text, which is placed below it. But in round 2, I wish to numerically calculate the magnification. Now, the major aim of this experiment is to notice how the magnification changes with varying parameters, and also to theoretically calculate the magnification and verify it with our practical results. In the first few slides, I have explained why the magnification takes place in the first place. So, due to a phenomena of light called refraction, light tends to bend away from the normal once it goes from an optically denser to an optically rarer medium. Due to this property of light, we find that a small object can be magnified to a larger image if a convex lens is kept above it. A water droplet is bulging outwards, so it also behaves as a convex lens. Now I will proceed to explain the two experiments that I have done. So the first experiment involves calculating the magnification with varying distances. The distance I'm talking about is the distance between our object and the lens or our imaging system. The setup is as follows. There are two decks of cards over which a thin plastic sheet is kept. Over the plastic sheet, I have a small droplet of water and a scale. On looking through the small droplet, we see a magnified image which I can measure with the help of the scale. And as I know the height of the object, the size of the object, the size of the image, I can easily calculate the magnification. After calculating the value of magnification for many different distances, I have obtained a table which I have also included in the PPT. But it would be better if I had expressed this tabular data in the form of a graph. The graph is also included in the PPT, and an interesting thing to note is that all of the points on the graph are extremely close to a graph of a more known function. The function I have given is 14 over 14 minus u. This ends our practical part for experiment 1. Now it's time to do the mathematical and theoretical analysis. After drawing the ray diagram for our water droplet and object imaging system, we find that the function which we had taken before, 14 over 14 minus u, is quite similar to the function we get theoretically, which is f over f minus u. Comparing these two formulae, we get that f should be approximately 14 millimeters. And after performing an experiment practically, we found out that f is actually 14 millimeters. So the theory checks out. This is a very interesting observation, and that ends experiment 1. The next experiment, experiment 2, explains how the magnification of our imaging system varies with the diameter of our droplet microscope. Now the setup is almost the same, but this time instead of varying the number of cards to change the distance, I fixed the number of cards in each deck and just varied the volume of water that I put into our droplet, which basically changes its diameter. Now, again, after taking various measurements of diameter versus magnification, I have prepared yet another table, and its graph is also included in the PPT. Now we notice that as the diameter is decreased, the magnification of the object increases, and vice versa. If I increase the diameter, the magnification decreases. The explanation for this phenomena is that as the diameter of our convex lens increases, the radius of curvature also increases, or you can think of it as becoming flatter. Now, after calculating, we find out that the focal length is actually directly proportional to the radius of curvature, which basically means that the diameter is directly proportional to the focal length. Now, as the diameter increases, the focal length increases, and as the focal length increases, we know that the magnification should decrease. Or as I have explained in the PPT, the magnifying power of a lens is defined as the inverse of the focal length. So obviously, as the focal length increases, its magnifying power decreases, and thus the magnification decreases. Finally, I wanted to explain the applications and our conclusions of this topic. We can use the principle we have just discovered to create a device which magnifies text, like text from a book. This would mean that we can replace reading glasses with something which is more handy. This tool, this tool must be made quite carefully, because we know that as the distance gets closer to the focal length, the magnification also increases. But at the same time, the image might also become hazier, which would mean that our device wouldn't be quite useful, would it? So we have to make sure that there should be an appropriate distance in our device which allows the object to be clearly magnified. The image should be clear, no haziness, and the image should be magnified. Also, there is another parameter which I haven't changed yet. The parameter of the refractive index. The refractive index parameter. For water, the refractive index is approximately 4 by 3. If I had used another liquid like glycerine, I could have gotten a higher refractive index and a higher magnification. So, varying these three things, the refractive index, the diameter of our lens, and the distance between the object and the lens, we can get our desired magnification. Finally, to conclude this topic, I would like to explain the entire PPT in a nutshell. Firstly, the object which is placed under our imaging system gets magnified. Secondly, as the distance between the object and our imaging system gets closer to the focal length, the magnification increases drastically. Also, as the diameter of our drop increases, the magnifying power of our imaging system decreases. For certain parameters of our lens, we can get a desired value of magnification. I really enjoyed conducting these experiments, and I hope to see you in round 3.